Dear students, please note down. These notes are available in the description of video. Please download your notes. Dear students, in today's lecture we will discuss Krebs cycle, or it is TCA cycle, tricarboxylic acid cycle, or it is also named CAC, citric acid cycle. As in this chapter, plant respiration, two important topics. One is glycolysis, second is Krebs cycle. In our last lecture, we have studied glycolysis. Means partial breakdown of this glucose molecule into pyruvic acid. This 6-carbon compound glucose is converted into 2 molecules of 3-carbon pyruvic acids. This Krebs cycle, it occurs in mitochondrial matrix. Dear students, while this glycolysis that occurs in cell cytoplasm, cytosol, because the enzymes required in this glycolysis are present in cell cytoplasm, while the enzymes for this Krebs cycle are present in mitochondrial matrix. This entire process, Krebs cycle, TCA cycle, it was discovered by Sir Hans Krebs. So it is named Krebs cycle. He described this entire process in 1937 and he was awarded Nobel Prize in 1954. The pyruvate or the pyruvic acid, the end product of glycolysis. This pyruvic acid undergoes oxidative decarboxylation. It removes one molecule of CO2. It requires NAD plus, nicotinamide adenine dinucleotide, and this NAD plus is converted into NADH2, or it may be written as NADH plus H plus. This entire process is catalyzed by pyruvic dehydrogenase complex enzyme. Dear students, the entire complex of enzymes, that is pyruvic dehydrogenase complex, in that complex Mg positive 2 ions, TPP, coenzyme, thymine, pyrophosphate, lipoic acid and trans-acetylase. And this step is, it is named gateway step, it is named link reaction, or it is transitional reaction, because it is a link between this glycolysis and Krebs cycle. That's why it is a gateway step, or it is a link reaction, or it is a transitional reaction. In this link reaction, this pyruvic acid is converted into acetyl coenzyme A. It is a sulfur containing acid, that is acetyl coenzyme A, and this acetyl coenzyme A functions as a substrate entrant for Krebs cycle. Note down, the glucose 6-carbon compound in glycolysis that undergoes partial breakdown. As a result, this 6-carbon compound glucose is converted into 2 molecules of 3 carbon each. These are pyruvic acid. And now pyruvic acid undergoes oxidative decarboxylation to produce acetyl coenzyme A. This acetyl coenzyme acts as a substrate entrant for Krebs cycle. And this step is, it is gateway step, or it is link reaction, it is transitional reaction between glycolysis and Krebs cycle. This entire process involves pyruvic dehydrogenase complex in which Mg positive 2 ions, TPP, thymine, pyrophosphate, lipoic acid, transacetylase enzyme are involved. This 3 carbon compound, it removes 1 molecule of CO2. It means this 3 carbon compound is converted into 2 carbon molecule, that is acetyl coenzyme, and it produces 1 molecule of NADH2 or NADH plus H plus. This step is gateway step, or it is a link reaction between this glycolysis and Krebs cycle, and this acetyl coenzyme A acts as a substrate entrant. Now Krebs cycle operates. It means the first step of the initial product of first product of this Krebs cycle is citrate, or it is citric acid. Now down in this Krebs cycle, the initial product, the first product is, that is a 6 carbon compound, because this 2 carbon compound acetyl coenzyme A in presence of this 4 carbon compound oxaloacetate. 4 carbon, 2 carbon, and the product is 6 carbon compound, that is citrate or citric acid. Now down, this citric acid is first product. It is initial products. That's why this Krebs cycle is named citric acid cycle, or it is CAC, or it is TCA cycle. Means this citric acid having 3 carboxylic groups. So it is a 3, or it is tricarboxylic acid. This citric acid is tricarboxylic acid. So it is TCA, or it is citric acid cycle, CAC cycle. First step of this Krebs cycle, that is formation of citrate, that is first product, that is initial product, that is citric acid. And this citric acid is formed by acetyl coenzyme A, that is a 2 carbon compound which interacts with this 4 carbon compound oxaloacetate. The reaction is catalyzed by citrate synthetase in presence of water molecule. That is, the product is citric acid or citrate. Second step of Krebs cycle, that is formation of cis-aconitate. Now down, the second step is formation of cis-aconitate, again a 6 carbon compound. It requires an enzyme, that is iron containing enzyme aconitase, and this aconitase converts this citrate into an isomeric form, that is cis-aconitate, which is 6 carbon compound. Third step is formation of isocitrate, in which this cis-aconitate, a 6 carbon compound, in presence of this iron containing enzyme aconitase and in presence of water molecule, it produces isocitrate, that is again an isomeric form. Fourth step is formation of oxalosuccinate. Now down, fourth step is formation of oxalosuccinate. In this step, this isocitrate in presence of this enzyme isocitrate dehydrogenase, and the reaction is catalyzed by NAD plus to produce NADH2 or NADH plus H plus ions. This oxalosuccinate, which is unstable compound, immediately this 6 carbon compound, it removes 1 CO2, and this 6 carbon compound changes into a 5 carbon compound, that is alpha ketoglutarate. Means the fifth step is formation of oxalo alpha ketoglutarate. That is a 5-carbon compound, because this unstable oxalosuccinate, it removes one CO2. The next step, sixth step, that is formation of succinyl coenzyme A. This step, succinyl coenzyme A, in which this alpha ketoglutarate undergoes oxidative decarboxylation. It means this step again removes one CO2. As a result, this 5-carbon compound changes into a 4-carbon compound, that is succinyl coenzyme A. And this entire reaction is involved alpha ketoglutarate dehydrogenase complex. That complex consists of Mg positive 2 ions, TPP, lipoic acid and trans-acetylase. In the presence of this enzyme complex, this alpha ketoglutarate 5-carbon compound is converted into 4-carbon compound, that is succinyl coenzyme A. In presence of this NAD plus, it means this step again produces NADH2 as well as it removes CO2. This is oxidative decarboxylation of this alpha ketoglutarate to produce succinyl coenzyme A. Step number 7, formation of succinate. Formation of succinate. In this coenzyme A is removed and this enzyme succinyl synthetase, it converts this succinyl coenzyme A into succinate, 4-carbon compound, and it produces one ATP molecule direct at substrate level. Note down step number 8, that is formation of fumarate. In this step, this succinate in presence of this enzyme succinate dehydrogenase produce a 4-carbon compound, that is fumarate, and this step produce FADH2. Means in presence of this FAD, flavine adenine dinucleotide, it is converted into FADH2. Each FADH2 yields 2 ATP molecule and each NADH2 or NADH produce 3 ATP molecules. Next step is formation of malate, formation of malic acid, in which this fumarate, that is a 4-carbon compound, in presence of this enzyme fumarase and water to produce malate. And the last step, that is formation of oxalosuccinate, oxaloacetate. In this last step, formation of oxaloacetate OAA, this malate, that is a 4-carbon compound, in presence of this malate dehydrogenase, means again one molecule of NADH2 is formed and each NADH2 produce 3 ATP. One FADH2 produce 2 ATP. It means in this entire process, one NADH2, second NADH2, third NADH2, 4 NADH2. 4 NADH2 are formed in one Krebs cycle. But dear students, here the products of glucose are 2-pyruvic acid. It means this Krebs cycle operates 2 times. Means all products become doubled. 4 NADH2 are doubled, means 8 NADH2 are formed. And 1 FADH2 in one Krebs cycle and in 2 Krebs cycles 2 FADH2 and 1 ATP in each Krebs cycle, but these Krebs cycles operates twice. 2 Krebs cycles operate because 2 pyruvic acid molecules are there, means 2 ATP are formed. The net equation 1 pyruvic acid plus 4 NAD plus FADH2 plus ATP. 4 NADH means 4 into 1 NADH equivalent to 3 ATP. 4 into 3, 12 NADH. 12 ATP. 12 ATP, 1 FADH2 equivalent to 2 ATP. 12 plus 2, 14, 1 direct, 15. Each Krebs cycle yields 15 ATP molecules. But as we know, pyruvic acid are 2 molecules. Means in 2 Krebs cycles, 15 ATP plus 15 ATP, 13 ATP molecules are formed. And in glycolysis, 8 ATP molecules are formed in glycolysis. Means complete breakdown of glucose produce 30 plus 8, 38 ATPs. The net equation of that, 1 glucose molecule in presence of 4 ADP plus 4 PI, 4 NAD and 2 FAD to produce 6 CO2 molecule. 1 glucose molecule is converted into 6 CO2 as each pyruvic acid produces 1 CO2, second molecule of CO2 and third molecule of CO2. As 2 pyruvic acid means 3 CO2 in 1 pyruvic acid and 3 CO2 molecules by second pyruvic acid. Total 6 CO2 molecules are produced from 1 glucose molecule. 4 ATP molecules direct, 10 NADPH, 1 NADPH produce 3 ATP, 10 means 30 ATP and 2 FADH2. 1 FADH2 produce 2 ATP, 2 into 2, 4. 10 means 30, 2 means 4s, 30 plus 4, 34 and 4 direct, 38 ATP molecules from this entire breakdown of 1 glucose molecule. 8 ATP from glycolysis and 15 ATP from 1 Krebs cycle as 2 Krebs cycles operate. So 15 plus 15, 30. 30 ATPs from 2 Krebs cycles and 8 ATP from glycolysis. Total net gain of 38 ATPs. Dear students, this is all about Krebs cycle, TCA cycle, tricarboxylic cycle, because this first initial product is citrate citric acid. So it is CAC, citric acid cycle, and this citrate citric acid having 3 carboxylic groups. So it is TCA, tricarboxylic acid cycle. That was discovered by Sir Hans Krebs. So it is Krebs cycle. Thank you.